Hello, everyone. Welcome to Invisible Worlds. When a crime leaves no witnesses, no cameras, and no obvious clues, there is one silent witness that never lies. That's DNA. And thanks to a technique called STR analysis, law enforcement can catch criminals with astonishing precision. But how exactly does this microscopic evidence help solve real world crimes? Today, let's dive into the fascinating world of DNA forensic analysis. Every cell in our body carries DNA, a long molecule made up of four chemical letters A, T, G, and C. This DNA encodes everything about you from your eye color to your blood type. In fact, there are 3 billion letters making up your entire genome. But here is something surprising. Not all of that DNA codes for traits or proteins. Scattered throughout your DNA are regions that don't code for anything, like little biological doodles. Some of these regions contain short sequences that repeat over and over again. These are called short tandem repeats or STRs. For example, a simple sequence like AGAT might repeat 5, 10, or even 14 times. Here's the secret: the number of repeats is different for each person. That means to identify someone, you don't have to sequence all 3 billion letters. You just need their STR profile. But what is an STR profile? Humans inherit one set of chromosomes from each parent, one from mom and one from dad. That means you inherit two STR numbers at every STR location, one from each parent. Scientists focus on 20 specific locations spread throughout our DNA called loci, places where STRs are known to vary widely between people. At each location, you have two numbers representing how many repeats you inherited from each parent. For example, at one STR location, you might have inherited 10 repeats from your dad and 36 repeats from your mom. I might have inherited 24 repeats from both my parents. That's also possible. Those repeat numbers for all 20 markers form your DNA fingerprint. Even close relatives like siblings have different STR profiles unless you are an identical twin. We have to understand something important. Each STR location in your DNA has its own unique pattern, like AGAT or TCTA, and its own range of possible repeats. For example, one STR might repeat 8 to 12 times, while another could range from 6 to 25. And because you inherit one version from each parent, your combination of repeat counts is completely unique. That's what makes DNA fingerprinting so powerful and so precise. So, how likely is a match? Matching one STR loci isn't that impressive. Lots of people might have 14 and 10 repeats at a given spot. But when you match all 20 markers, the probability of that happening by random chance becomes astronomically small. So, how small? About 1 in trillions of trillions. And 1 trillion has 12 zeros after it. In short, it is virtually impossible for two unrelated people to have the exact same STR profile. Let's get back to the crime scene. Once a STR profile is created from crime scene evidence, it can be compared to either suspect's DNA if there are any suspects, or DNA stored in criminal databases like CODIS, which is the Combined DNA Index System, or even distant relatives through genealogy databases, which people use these days to trace their family history or know more about their ancestry. But of course, DNA forensic analysis isn't magic. There are important challenges. Contamination, mishandling evidence can ruin a DNA sample, and mixed samples. When DNA from multiple people is present, the analysis becomes even more complex. And then there are ethical debates, especially when using relatives' DNA without their consent. But when done carefully, STR analysis remains one of the most reliable and powerful tools in modern forensic science. DNA forensics isn't about reading all 3 billion letters. That would take forever. Instead, scientists focus on just a few spots where our DNA tends to vary. Even though we share over 99.9% of our DNA, these tiny differences are powerful. With just 20 markers, investigators can build a DNA fingerprint strong enough to solve crimes even decades later. It is the genius of using less to reveal more. Every cell, every trace, every fragment tells a story. And now we have the tools to listen. Thank you for hanging out with us today. Until next time, stay curious.